Hello everyone. So as a part of this introductory neuroscience and the neuroinstrumentation course, I will be giving the next demonstration about P300 using the EEG lab and ERP lab analysis. So the previous two tutorials I have been giving based on the MMN and the auditory evoked potentials. Now I will just give you an overview on background about what is this P300 about and how does the waveform look, and then I will be giving a small detail about how does the demonstration been carried out. So it is a bit different from the MMN and AEP again in the the event list, how it has been created and some operations and all that has been done, how to visualize it and everything. So what is this P300? So the P over here, it represents the positive peak that has been obtained, and 300 means it is actually the it's like the positive response that has been obtained in the 300 millisecond in that range. So it will be obtained always in that 300 the 300 to 600 in that range it should be obtained. So this is about the P300. So always, as I told you, there will be a wet electrode, we have to peak, it has been to be put in the head, and then there should be a contact between the electrode and the system so that we can obtain the scalp potentials. So how is it, how the experiment is being conducted is that there are basically three stimulus in this. So first is the standard stimulus that has been that is usually being given, and here we are, and P300 is about the visual how it looks, how do we perceive whatever we see, that is how it has been recorded. So there are two components that will be there in this P300. One is this P3A and the P3B. There are two components that has been obtained. So one is the brain. It should be able to check how the how the brain is able to attend, how it is being able to give attention to what we see. Like for example, now if we have, there is a difference in the two. So if we have a 4 centimeter ball and a 6 centimeter ball, our brain should work in such a manner that it should be able to differentiate the 4 centimeter and the 6 centimeter. So that is what is done by the attention. That is, it's done using the attention, how we perceive it. So it based on that, it is the how that is how the P3A works, and P3B is something that is based on the how the memory is working. So even if we have to differentiate between these two, we have to there will be another one stimulus also, like this checkerboard or something, like a distractor will be presented in P300. So what will happen is, when this distractor is being given, we have to still remember that okay, the 6 centimeter ball is the difference and 4 centimeter ball is also different. So that difference also you should remember, and we have to pay attention also, and we have to keep it in the memory also. So those two parts of the brain is being studied in detail using this P300 analysis. So both the working memory as well as the attention seeking memory of the cognitive brain, how it works, that is being explained, or that can be studied in detail using the P300 analysis. So here, as I told you, there will be three stimulus basically. One is the standard. The standard, it can be the normally maybe in this experiment which we have conducted over here, we have used a 4 centimeter blue colored ball as a standard. So and then we will be giving a distractor as well as a target. So these three are the stimulus that has been given, and about the experimental details, as I've mentioned, it's all the same, the Neuroscan EEG system, where we have the 64 electrode channel, and then we use we have before acquiring the data itself, we have to have the band pass filter, the notch filter, and all the artifact rejections and all to be done has been performed. And the P300 mainly it has been obtained from the frontal as well as in the temporal parietal areas of the brain. So in the next figure I will just give you a brief about what are the different stimulus, and how does the where does the where do we get the brain, in which part of the brain we are actually obtaining these P300 responses, I'll be saying. So here the stimulus here there even in P300 also there are various different types. Now what we had performed is the visual P300. Even there is an audio P300 also, wherein we have a target and a standard. In there we don't have a distractor, but we will just have a standard and a target. So in visual we have three stimuli. One is the standard, which like for example, if we have 1,000, totally we want 1,000 stimulus, means over that the 800 stimuli will be of over 1,000, 800 stimuli will be of this 4 centimeter blue ball, the small sized ball. So that will be just like the responses which will give the N1 P2 P1 P2 N1 complex, that particular thing it will give. It's just to sense, it's to just check whether our visual cortex are all working properly and all. So that is for this standard. Next is this target. Target is actually a 6 centimeters blue ball. So here in this experiment what we have to do is that we have to instruct the subject in order to give a response to the target. So whenever there is this particular experiment has been running in that place, we will have a response pad being given to the subject, and whenever there is a target, that is the 6 centimeter blue ball, whenever we are whenever we encounter this ball, the 6 centimeter ball, we have to ask them to give a response. So this is about the that is about the experiment, and this it will say that okay, the brain will differentiate, okay, the 4 centimeter ball and there is a 6 centimeter ball, and when there is a 6 centimeter ball we have to give a response. So all of this the brain will know and it will do it. So that is why we say that the attention memory of the brain is been working, and the attention as well as the working memory, both of them is been working in this condition. And there is a 10% of a checkerboard also been given, like it's just a chessboard kind of thing with this is how it has been given. So this will just it's like acting as a distractor. Whenever there is a this checkerboard comes, again we have to our brain will have to come to the normalcy that okay, there is a standard and this target, and now we have to differentiate again the 6 centimeter ball. So all of this working memory and the attention, everything together it works, and then this is how we get the P300 response. And in P300 itself there are two responses, one is P3A and P3B. So P3A it is like the earlier component, and the P3B is the a bit later component. So over here if I show you and another one important thing about this is it's all randomized. It's not like this MMN always there should be three that B boop or B boop like that. That is not the case in P300. It is always randomized. We have to give a percentage, like 80% I want the standards to be coming, 10% the targets, and 10% distracted. So whenever it's like a artificial intelligence, or it's some program that has been done by the stimulus system, and it will give it gives in a randomized manner. So in this condition, for example, there are first two standards, then a target, then again 3 standards, then a deviant comes. So it is all randomized. We wanted out of 1,000 response, out of 1,000 stimuli, 800 will be for standards, 100 will be for the target, and 100 will be for the distractor, and it will be randomized. It is not that always first 5 will be standard, then a target, then a distractor, not like that. It can be all randomized. That is the main important step, stimulus thing that you have to remember about the visual P300. And if you see the image over here, the P3A it comes in the frontal part of the brain, and the P3B it is in the parietal part of the brain. So frontal and parietal, and mostly the central parietal, all these areas are the most commonly activated during the visual P300 stimuli or the ERPs are being obtained in these parts of the brain. So when we are doing the ERP analysis also, we should concentrate more on those parietal regions or the central parietal temporal regions, and also in that areas only the P300 response will be maximum. So there is a positive, as I told you, at 300th millisecond, in that time we will have this P3A. It starts the P300 response. It is obtained from that particular thing. And here we have a response being, as I told you, there is a response that should be given for the whenever there is a target being seen. So these are the different stimuli, and this is how it actually been done, and the experiment, I mean the brain, how it looks and how from where it has been obtained. And then the demonstration part is the similar as that. I only think that here in this we have to concentrate on the target and the distractor, and then the P300 triggers based on that, and even the epoching also will differ, because we want it to be from minus 50 to like 600 or 650, like that, because our response in this case, it starts from 300 only. So that's why we take from minus 50 like a baseline, and then to the next level, like till 600 or 700, like that we will be epoching in this condition. Similarly, all other steps are similar, only the difference is in the event list and the epoching. And the plotting also, we can just give a difference, or just give however like for both the bin values we can directly do, or we can do even for the separate analysis also. And the P300, if you have to say about the papers that they have used, this Pollock's paper is the best paper for P300, because they are the ones, he is the one who actually started all the works about P300. So it's like a base paper for P300. So as I told you, along with this visual, there are auditory stimulus also been given for P300. So their topographic analysis, everything can be studied with these papers. Next, I will be speaking about the demonstration in the MATLAB using EEG lab and ERP analysis. Yeah, thank you.